So now I'd like to invite Miss Sreegauri Krishnan, Krishnakumar. She is going to help the students go through this entire experience of showing their posters that could be evaluated. Over to you, Sreegauri.
All right. Thank you so much, Dr. Mohit. So far, we had an exciting sessions that talked about the various active areas of research that are happening in the field of bioinformatics and data science in the academic industry in India. And we also discussed some of the what are the various opportunities that students can access and where to access these opportunities and how to access them. Keeping these in mind, there still remains one of the challenge that students face, that is they don't get the opportunity to showcase their project after completion. So that is why the Omics Research Symposium encourages students worldwide to present their research project and network with peers and experts in the field. So the topics of the symposium is Omics and Bioinformatics, AI, Machine Learning and Data Science, Astrobiology and Space Omics, Computational Biology in Biotechnology and Pharmaceutical Industry. The submission was open to all registered participants from high school, undergraduate, graduate, as well as citizen science projects. And out of the 25 plus abstracts that we have received, we have selected 12 of the finest projects for further rounds of review by the judges. Now, let me take a brief moment to introduce you to the judges who will be reviewing the student projects. First, we have Dr. Ramesh Kumar. He is the assistant professor at Reva University. We are also joined by Dr. Harpreet Kaur. She is the postdoctoral researcher at NCI NIH. We are also joined by Dr. Raghavendran Lakshminarayan. He is the research consultant at Fine Biotech. And Dr. Abhishek Sengupta is the assistant professor at MIT University. And finally, and last but not the least, Dr. Julia Panov. She is the associate director at Turbo Bioinformatics Research Center, Israel. The presentation will be judged in 4 categories, that is introduction, methods and results, discussion and conclusion. Finally, if the students are able to showcase a quality-driven project within the time allowed, the best posters will be selected on the following different categories, that is best technical research project, promising research direction for future potential, and most innovative approach in an established domain. And the guidelines for the competition are simple. The participants will be given 5 minutes to present their poster. Once the time is up, you'll have to stop your presentation and move to the next candidate. So kindly make sure that you complete your presentation within 5 minutes. With that, let me invite the first participant who will be presenting their project. That is Dr. Parak Sehgal. She is the professor at Rangta College of Science and Technology, Chhattisgarh, India. And she and her team will be presenting her project on characterization of the hypothetical protein sequence of Naegleria fowleri using bioinformatics approach. So over to you, Dr.
Pat. Yeah, ma'am, am I audible?
Yes, you're audible.
Ma'am, I'll be just a moment, ma'am.
All right, sure. Could you please share your screen?
Just give me a second. Ma'am, is my screen visible?
Yes, it is visible.
Okay, thank you so much.
Just your time starts
now. Okay, ma'am. Good evening to all the judges. Myself, Dr. Parak Sehgal, and I'm representing Rungta College of Science and Technology, Bhilai. And my colleague is Pratigya Das, Research Associate at the Biotech. We have chosen a topic to work on characterization of a hypothetical protein sequence of Naegleria fowleri using a bioinformatics approach. Now, this particular amoeba is causing a disease which is called as PAM. Okay, so PAM is primary amoebic meningoencephalitis. This disease is basically caused when somebody goes for swimming in the open kind of water environment. So this particular amoeba is inhaled via nostrils, and this can cause a lot of issues and can be very infectious and can be fatal for one person's life. So particularly when it is taken inside. Now there are three lives in the life cycle of Naegara floweri. There are three phases. So one is cyst phase, another is trophozoite phase, and there's a third one. So in these three phases, the trophozoites are the most severe one, which causes the infection because they can multiply themselves with the binary fusion method, and they can cause that much of infection that a person can die in 7 days. Okay, so while looking to this particular slide, we can see if cerebrospinal fluid, the diagnosis is cerebrospinal from the cerebrospinal fluid, that gray to yellow color is changed, and when 1 ml of blood sample is taken and it is being checked, so it is found that the red blood cells number is very high. So person can be suffering from this particular PAM disease. There are various type of treatments known for this. So this is a life cycle I've already told about cyst, trophozoite, and flagellate phase. So we can see here the division of the cyst to trophozoite phase, which is the infectious one. Okay, so in this particular project, I have tried to find some details about the hypothetical protein which is not available in the database. So I have tried to find all the information about it using various bioinformatics tools. And there is something new which I have discovered in the project while doing the calculations with the bioinformatics tools about Naegara floweri, which causes this disease. So we can see the list of the bioinformatics tools I have used. I will tell you what new thing which I have discovered when I was doing the project. So this is the approach I have used. This is the hypothetical protein, and this is the ID number of N floweri, that particular protein which about which the information is very less available in the databases. Its structure is not there, its detail is not there, its protein-protein interaction was not available. So by the approach, I was able to figure out about this. So this N floweri, this particular ID was, I have got this from the Uniprot database, and I have tried to perform the BLAST-P, by which I was able to figure out the list of 10 sequences similar to this particular ID, and that was easily available. And there were around 82 of the sequences, but I took the first 10 to understand what could be the maximum similarity with its co-similar kind of species from the same family. So the first 10 sequences I have taken N floweri 5, and the next thing what I was to tell you was that there is another species called as N gruberi. Okay, so Naegara gruberi. I have taken 5 sequences of that and 5 of floweri just for the supportive study. So after I generated the multiple sequence alignment of these 10 sequences N floweri and N gruberi, I was able to see the conservation of the amino acid pattern in the sequences that supported the data that they belong to the same family. And then I have tried to discover the kind of family they belong to, the same family. And there was some patterns, common patterns were seen in the ProSite database. And something which was different was in the string database. When I wanted to see about the protein interaction, I was not able to figure out anything for N floweri. But when I took the next 5 sequences of N gruberi, I was able to figure out some similarity in the co-similar sequences of N gruberi. So this was something new which is not yet reported. So I figured out this. And after the structural characterization, when mod base I have used, there was no structure which was modeled for N floweri. And I was able to make it with the help of Swiss automated modeler. The results I'll be showing soon. And more information about this particular sequence I have figured out in Amoeba database, which shows that this particular hypothetical protein is very important in some kind of KEG and metasec pathways, which I'll be showing later. So this is very much important in doing some metabolic activities. So we are going to the results section from the Uniprot. This is the ID I have retrieved for Naegara floweri. And this was all about the project, where this was the main idea to figure out the details of it. These are few sequences which I have taken for N floweri and gruberi together just for the supportive study. This is the multiple sequence alignment. So I can show you the conservation over here. The sequences are very highly conserved amongst these 10 sequences. And I was able to figure out from the PFAM and the CDD database that they are all belonging to have the HSP70 domain. So this confirms that it is also having HSP70 domain like rest of the sequences. Then from this particular analysis of CDT database, it confirms that they are belonging to the same clan, 36495. They're having the same accession number and the same superfamily, PTZ 4x09. Then from the Interpro databases, these are few of the patterns from the domains I was able to figure out. IPR numbers are given. So I was able to figure out that they are belonging to the same family and clans. Then I was able to figure out some patterns which were similar. So I can show you a heat shock protein, heat shock HSP70 patterns are present. So they are having around 5 sequences. When Floweri was there, they were around 15 hits I have got from ProSite. This is the string result. So this tells that when I have used N-Floweri, there was no result. And just to support my study, I have tried that if N-Floweri is not there, N-Gruberi can be present for protein-protein interaction, which is just similar and belongs to the same family. So I was able to see that there are co-occurrences. There are some literature-based reports available. These are those ID numbers which I've shown you in the BLAST results. So they are having some or the other relation between them. Then using the CISP model, I was able to figure out that with this template number of 4ANI1, it was able to generate based upon sequence identity that the structure can be like this for the hypothetical protein, which is not available in the database. And very much important, it is based upon sequence identity. It means that this model has been generated based upon the place of the amino acid. So this is based upon the sequence identity. Then
sorry,
using
Dr. Parekh, could you please stop up?
Yes, ma'am. Yes, ma'am. Okay. So this is the last result where I can show you the amoeba database has shown that these are some of the important pathways, like it is involved in the purine metabolism and thymine metabolism. So overall conclusion of my project is that whatever information was available for N-Floweri was very less to interpret. In India, there are only around 15 cases reported. And with the help of N-Gruberi data, I was able to support my studies that some information can be said that Gruberi and Floweri are similar. They belong to the same family, and the results can be checked that way. Thank you so much.
All right. Thank you so much, Dr.
These are my
Parekh.
references. Thank you, ma'am.
The judges, do you have any feedback to give to Dr. Parekh?
I'll just show my poster as well.
Judges, do you have any feedback to give to Dr. Parekh about her project?
Yes. Hello. Hello, Dr. Parekh.
Yes, ma'am.
It was nice work. So I have one actually suggestions maybe when you're choosing like 10 sequences. So
Yes, ma'am.
instead of choosing 10 sequences, it would be better if you use some criteria like maybe percentage of similarity. So maybe 90% similarity of the sequences. So maybe that would be the better criteria rather than just choosing the top 10 sequences. So because when we comparing some sequence, so we should consider its similarity or dissimilarity score. So my suggestion is fine for this work if you're just doing for the publication perspective. It would be much better like if you consider that. Yeah. Rest of thing is fine. Like it is interesting work. Yeah.
Okay, ma'am. Thank you so much for the suggestion. Ma'am, just I would like to say something because the idea behind this project was not to figure out the similarity initially. It was just about to figure out only the information about the hypothetical protein. So that criteria I may have missed as per you have said. So, but yes, for the publication point of view, for the next, I will consider this. Thank you so much for the suggestion. Yes, ma'am.
All right. Thank you so much, Dr. Parekh. Could you please stop sharing the screen?
Yes, ma'am. Yes.
Thank you. All right. So let me invite the next participant, Mr. Kashish Kundu. He is a postgraduate student of bioinformatics and computational biology from Amity University. And he'll be presenting his project on in silico analysis of transcriptional responses to UV and IR radiations. So over to you, Kashish.
Thank you so much, ma'am. Yes. So good evening, everyone. Myself, Kashish Kundu, and about my poster. In skin cancer, many genes are involved in known DNA damage response pathways for cell cycling and proliferation, and also in apoptosis, the DNA repair or in other stress responses. However, majority of these genes are involved in unexpected pathways with functions in signal transduction, RNA binding and editing, protein synthesis and degradation, energy metabolism, and cell structure. I was interested in knowing these responses, hence I chose a particular dataset to work on. The analysis was on the dataset was done on T-Bioinfo server along with a few other tools and platforms. This part of poster represents the, wait a second. Yeah. So this was the introduction which I gave about my poster. Yes. So this part of the poster represents the methodology that was used along with a flowchart illustrating the pipeline that was followed. Firstly, the dataset was identified on NCBI using GEO dataset database. The database was downloaded and filtered, and grouping of the sample was done. Of the samples was done. PCA and other statistical analysis were performed manually as well as using GUI platforms. Later on, pathway analysis was done using Links tool and PantherDB. Links database, that is library of integrated network based cellular signatures, takes up input as gene IDs, fold change information, and an optional field for p-values. Protein analysis through evolutionary relationships, abbreviated as Panther. It links protein sequence evolution to the development of protein activities and biological functions. Separated files of genes are accepted as inputs, and as output, graphical representation of gene expression levels are plotted against the pathways they are expressed in. From here, further biological interpretation was made. From the top probe sets, the IR responsive genes like ATF3, MAP2, MEF2C were involved in stress response. Genes like PVT1, MMYC, NEK2 were involved in cell cycle and proliferation. There were many genes with involvement in signal transduction and apoptosis. For UV responsive genes, similar results were found with entries in stress responses, signal transduction, energy metabolism, and apoptotic pathways. From the results recorded, it can be interpreted that abrupt activities of cell cycle and energy metabolism may cause IR as well as UV-induced skin lesions. Apoptotic cycle showed an increase in the expressions, which can be inferred as immune activity to tackle the lesion development, or it can also indicate the breakdown of essential cellular components that were aided in prevention of further proliferation of abnormal cells. The outcome proposes that protein translation is additionally hindered by halted transcription of tRNA synthetase genes. Now, here you can see the PCA result, which was drawn out, and this shows the PantherDB result along with the bar charts of the genes that was expressed. Now, using PantherDB, these two tables shows us the threshold that I took, that is the 9 gene from this table, and I studied these pathways. I haven't covered all these pathways because instead of going into an extensive research, I was more focused on the highly expressed genes, and hence I went for these pathways.
Kashish, sorry to interrupt you. You have less than 20 seconds now.
Okay, yeah, I'm concluding. So the conclusion section gives us a brief overview of the interpretations of the analysis, and the bar chart represents the probe set information of induced and repressed genes of IR and UV. And for future perspective about this project, now I'm looking into the method comparison of this data set that was that I chose along with the method that I used, and I am looking forward for its validation now. Thank you, ma'am.
All right, thank you so much. Does the judges have any feedback for Kashish? Dr. Raghav, Dr. Harpreet, do you have any feedback?
Hello, Kashish. So I have just a few things. So it would be better if you just also show like how many samples have you used. Maybe I missed that. So that it should be like statistically correct. Another thing, when you actually this is just for the I mean from the analysis perspective. I'm not going into the deeper science. So another thing in your principal PCA results and your pathway results. So it would be better if you maybe do it based on the sample group, because when you're drawing this PCA, it is difficult to understand like what is actually which group is there or which is not. I guess you just assigned it
No, I did.
or something. Yeah,
Ma'am, actually, before I have the result for both before grouping the PCA, actually I was looking if there is any similarity or any plot interpretation which can I take, which I may find without grouping the data. Once it was grouped into IR, UV, and separately for UV and IR, I have inferred that information also there.
so it would be much better when you're doing even in the first group before grouping. You can take actually, I mean, you can color your sample based on either this radiation type, because it's difficult to interpret anything out of this, like whether these groups are distinguished from each other or not. So that's something that you need to maybe must do this or reanalyze it. So because it is difficult to interpret at the moment. Yeah.
Okay.
Yeah.
All right,
yeah, that's all.
All right then. Kashish, could you please stop sharing the screen?
Yes, ma'am. Thank you, ma'am.
All right. So next, I would like to invite Mr. Emmanuel and team who will be presenting their project on visualization of the chromosome 1 genomic data from the African, American, Asian, and European populations. Over to you. Emmanuel, are you there? All right, then. So let's move on to the next participant, Ms. Anmika Ghosh. She will be presenting her project on transcriptomic analysis of hippocampal subfield CA1 in Alzheimer's disease. Over to you, Anmika.
Hello all. So I'm sharing my screen. Is it visible, everyone?
Yes, it is visible. Could you please zoom in? Or thank you so much.
Yeah, so I can easily zoom in here. Okay. So hello everyone. So today I'm going to present my project topic, transcriptomic analysis of hippocampal subfield CA1 in Alzheimer's disease. So what is Alzheimer's disease? Alzheimer's disease is a neurodegenerative disorder affecting mainly the elder population. While it could be observed in the younger population, but its prevalence is more in the older generation. There are several hypotheses that exist behind this Alzheimer's disease. However, some of like not all of them are really accomplished or really found enough evidence. So here is a comparison between the healthy brain and a brain that is affected by severe Alzheimer's disease. And here is another diagram that shows hallmarks of Alzheimer's disease, which include accumulation of amyloid plaque proteins, neuroinflammation, mitochondrial dysfunction, autophagy dysfunction, and cholinergic insufficiency. And last but not least, a neurofibrillary tangles. So here is an overview of the methodology of what we have done. So for the sake of project, we have used this data set from gene expression omnibus. The pre-processing of the data was carried out with the help of Microsoft Excel 2019. After pre-processing of the data, we have quantum normalized our data and labeled it. Followed by this, we done principal component analysis, and we detected X119 as an outlier and removed it. Then we ran DSIC2 and insert enrichment analysis. After determining statistically significant genes, we validated our results. So here is an overview of like our results section. So as we can see here, this is the result of the principal component analysis. So the PC1, it accounts for 23.01%, PC2 accounts for 18.19%, and PC3 accounts for 14.75% variance. X119, as mentioned earlier, is detected as the the outlier, and we have removed it and proceeded further. So here is a volcano plot showing the upregulated and downregulated genes, and the right side shows the positive upregulation and upregulation, whereas the left side shows the negative upregulation. Here is the gene ontology analysis for our data set. So activated and suppressed pathways are separated by two separate boxes, and the gene count is reflected by the size of these circles. Gene ratio is mentioned at the bottom of the boxes, and here is the mention of the p-adjusted values. And this is the CNET plot of our genes and gene ontologies. So both these are the genes that are, the fold change is negative, so they are basically down regulated, and there are some gene ontology terms, again they are also down regulated. Here is here is the result of our cage enriched pathways analysis. So here we can see again that activated and suppressed pathways are separated by two boxes. Gene ratio is mentioned here in the bottom, and here is the like we can see the p-adjusted values. The red signifies the most significant ones, the statistically significant ones. Okay, so for the sake of the validation, what we have did is we took the gene list of the table supplementary table S5 of the main paper that is associated with our data set, and we compared it with our results, the output file that we got after DSIC2 analysis. So we mainly went for the statistically significant values, statistically significant ones. So our results shows that there is 0%, there is no overlap between this two list of genes. So therefore we concluded that that it might be due to the fact that our project is very much focused on the hippocampal subfield CA1, whereas the main paper was taking into consideration other hippocampal subfields. So that is what I can say about the conclusion section, and here is my references. And last but not least is I would love to acknowledge my research mentors, Mr. Dr. Raghavendran Lakshminarayan sir and Elia Brodsky sir, for helping me out to carry out this research project. I would also love to thank all the members of the Pine Biotech. Thank you.
All right, thank you so much. Does the judges have any feedback for Anamika?
Yeah, I can ask this time maybe. Thank you very much for the presentation, and just a few questions about your presentation. So you mentioned that there was an outlier in your data set and that you excluded it. So did you try to find out and understand why it is an outlier? Did you go back to the data and try to understand why this sample is an outlier? Maybe some normalization was done differently, because the main paper, as I understand, did not mention any outliers, correct?
Yeah, the main paper. Actually, to be honest, I back searched about the sample. It's 119, but I could not find anything really significant to tell actually. But that's I could not find anything.
Okay, yeah, just remark that usually an outlier is something that you should probably go and really dig into and try to understand why it is an outlier. Maybe the problem is not with the sample, but with the analysis. So maybe, analyzing and looking at the data a little bit differently would yield different results. So also you mentioned the downregulated genes and upregulated genes, and the pathways and everything those graphs that you talked about. What I'm kind of was a little missing is your interpretation. So are those pathways or genes mentioned in literature? Have they been found in Alzheimer's disease before? Some of your input on the interpretation of the results. So that was a little bit missing for me. And also, the last graph where you have the Venn diagram and you show that you have nothing actually in common with the main paper. Maybe I didn't, I didn't have time to go into the data set itself, but you probably took only part of the data from this big data set, I'm guessing, right?
Actually, we have considered the statistically significant genes only, that is p-value less than 0.05. So it was mentioned in the supplementary table S5 in the main paper, that is the last one, and here is actually my 181 gene. So I compared that.
Right. So you said that none of the genes actually were the same. So I was wondering, probably they had more samples and maybe did different differentiation, or was it the same? Because if you had nothing in common, that's a little, concerning, I guess. that's just interesting.
Actually, I take it in another way. That paper, the original paper, what they did was related to all hippocampal sepals and also EC or entorhinal cortex. But when I did not find any overlapping gene, I went back to the data, the GSC file, and what I found out that the data submitted in the GEO or Gene Expression Omnibus database, that is very specific to hippocampal sepal CA1 that I have mentioned. So I am very positive about my results actually, because I think my result is specific for CA1.
Okay, no problem. Just wanted to.
Can I interrupt? Actually, I was expecting Animika to tell this. And the comparison that she did was with the differentially expressed proteome from proteomics data analysis from different regions of Alzheimer's disease patients. So that's what the paper actually listed, and that's what she took to compare her differentially expressed genes list that she got from hippocampus. That's why she did not find a lot of common genes between the differentially expressed proteins analysis from proteomics data, which
I see. This is proteomics. Okay.
Yeah, averaged out data from several regions of Alzheimer's disease patients from one specific region that is hippocampus. That is differentially expressed genes that she found out.
I see. Yeah, and I think this is pretty much known that there is little, actually, a lot of times there is little interaction between the proteomics and the gene expression. So yeah, this makes sense. Thank you.